I feel a little weird about this only because this is a little different than what I normally do here. On Monday, we went over the dual oscillator paper, or dual oscillator, the dual model, CAM plus model, hybrid model. That was it. And I just felt like I didn't really understand some things deeply. So I decided, I asked Florian for, what would be a great paper to read, and he suggested this one for Burgess, Barry, and O'Keefe, which is basically about the oscillatory inference model. So I read this carefully yesterday. I'm still confused about things. There's some fundamental ideas in here that I'm struggling with. And so what I would normally do is I would spend several more hours or a good part of a day reading a bunch of references in here. But I'm being a little bit lazy and I'm trying to get shortcut things here. So I want to just talk about problems that I don't understand. And rather just have a discussion, if someone can explain it to me, that would be helpful, or maybe I'll be exposing some things. So it's sort of atypical, and if anyone else doesn't want to participate in this, if you want to go do something else, go ahead and do it. I just have questions that I just would like to pose and see if anyone in the room knows the answer to those questions. So I really only expect a few people might, Marcus, Subotai, and myself. Well, I don't have the answers because that's why I'm asking the questions. So it's a bit unusual. So if you don't want to hang around for that, that's what I want to do today. I want to go through some issues in this paper I just didn't understand. Is that okay?
Yeah, fine with me. I have some grid cell related questions too, but they're more general and they go in a slightly different direction.
Okay,
so
start
with that.
I'll start with this. I'm going to give a very short sort of this sort of historical viewpoint as I read in this paper. Just to provide the context of this, because I think that's important. It somehow feels important to me, like how did this idea of the oscillatory inference model come about? And maybe you already talked about this, Florian, I can't remember. But the basic idea of this oscillatory inference model did not come about to explain grid cells. That's not what it was about at all. It was about explaining a particular observation about place cells, right? And this is the sort of the phase shifting of a place cell. So a place cell fires as you enter into the place it's encoding. It starts firing more rapidly, and as you exit, it fires less rapidly. So it's sort of this scalar encoding. But what they noticed that there was this phase shift, right, where the cells, the firing of the cells. So if you're going through this space here, and the animal's moving like this, and as it's moving through here, you start getting some spiking, and you get more and more spiking, and then it diminishes again. But they found out that they observed that there's this background theta frequency that's going on, and these spikes occur relative to peaks in the theta frequency. When you're in the middle of the space field, you see the spikes all bunched around like that, but when you're out to the edge here, they get shifted. So I believe, I can never remember which one. I think they get shifted. This is late here, and this is early here, right? Right. Yeah. So late meaning would be spiking here, and early meaning would be spiking there. And they argued that the phase shift is a much more accurate indication of where you are than the actual spiking frequency, which is an interesting thing because they argue that's really the encoding, which that's one of my questions I'm going to come back to in a moment. Because I don't understand how we incorporate the encoding.
And how many times does it spike within a single cycle?
They didn't say that in this paper. They showed little cartoon drawings where they would show 2 spikes and 1 spike.
Right. So the typical recordings that I've seen, it's a limited number of spikes you get passing through a place cell or a grid cell field. And typically, like the total spike count is somewhere between 2 and 6 or something.
Total number of spikes?
Yeah, it's actually quite
within
low.
a cycle, or I'm talking about within one of those cycles.
No, all the way passing through that.
That means a bunch of these cycles it wouldn't be spiking at all.
Yeah.
That implies it because these are about a tenth of a second.
Right.
And therefore, if it takes a second or two to go through here, you're going to go through 10 of these phases, and I'm only getting 2 to 4 spikes, and most of these peaks I'm not going to get.
So typically, you stay less than a second in the firing field. So typically, it's maybe it's 4 theta cycles or something, and you might see 1 spike on the first and 2 spikes on the second.
Now, the big question here is, will there be some peaks where we don't see any spikes? Is it interesting?
Yeah. Sometimes you're also missing. Sometimes you're missing the entire field. That also happens, right?
So basically, they were trying to explain this phenomenon. Why there would be a shift here. That's all they were trying to explain initially. And the answer is in some sense in hindsight obvious, but it's a very good one. It's almost certainly the correct answer. Is that this is the kind of behavior you get when you have two frequencies that are slightly different, two cycles that are slightly different frequencies, and if you add up those two frequencies, you'll see that the peaks occur here. The addition or the multiplication of those peaks would occur here and here, and the size of those the additive of those two cycles would also indicate the number of spikes you got. So this kind of interference pattern is common. I'm familiar with the tuning of pianos. It's something you have to know about in tuning pianos. So it's just I'm very familiar with that, and I said, oh, that's got to be right. That's it. That's the right answer to explain that phenomenon. Then they said, okay, well, that wasn't to explain anything else. It was just to explain this phenomenon. And then they said, well, and only for place fields. And then they said, well, there's a problem because and then they're saying, oh yeah, well, they're saying there's going to be one of these is going to be some sort of background theta. And then this other one is going to be some sort of a cycle on a dendrite, a dendritic theta-ish cycle. And this one would change. It would be a voltage controlled. They just assumed it's voltage controlled, but it basically would reflect speed. So how much this shifts from theta, so this gets faster or not, depending on how much this shifts from theta, is depending on the speed of the animal, and that would explain why the animal goes through this quickly or slowly. You get the right answer there. So that was just, there's a lot of stuff, and they came up with this ridiculously simple, it's a good idea, but it's basically the model of the neuron is that you have a dendrite and you have a soma, and they have two different frequencies, and this one's going to be speed modulated.
Right, so it's a little bit faster.
A little bit faster on speed. Of course, that's not what neurons look like, but that's the idea they came up with. But the idea that you have two oscillations is the right answer. And I'm not sure if this is where the two oscillations are, but that is the right answer, it seems to me. Anyway, then they noticed that, well, this is a problem because in the place field, if you had this mechanism, the animal kept moving, then you would expect to see another place where it activates, another place where it activates. And they said, well, you don't see that in place fields, so we have to come up with a way of preventing that from happening. So they proposed this idea that there would be that when you were out of phase, and I think it was that they locked them out of phase or something, they came up with some mechanism to prevent this from occurring. Of course, then along comes the idea of grid cells, and this is exactly what you see in grid cells. Like, oh, hallelujah! Then we can accept that. Grid cells do this thing, and that's what these things are becoming in and out of phase, as slowly as being modulated to envelope. And so they said, great, we're going to use this model to explain grid cells, and because it really doesn't explain place cells. And then so they started talking about grid cells, and then of course. There's multiple problems with this because we needed to have this directional thing. So then they proposed this idea of 3 different 60-degree oscillators in the cell, the model we talked about. Yeah. So that was the history of this, and it was complex. I had some really basic questions about this. The first one, there's this idea that there's an oscillator in first of all, there's a background theta oscillation. So that's like a background field potential, I guess, they measure. Where does that come from? Do we know that just doesn't exist. There has to be neurons that make that happen, and they have to have some way of affecting a large amount of tissue. And first question, does anyone know how that occurs? Are there cells that distribute throughout the hippocampal region that are just putting out, I don't know, modulators everywhere? I mean, how does that come about? Does anyone know the answer to that question?
I think I do.
Okay, good.
I'm not an electrophysiologist. I'm not an experimentalist. I've not.
All right, enough. I'm not any of those things either.
Right.
I'm not even a neuroscientist, and I'm afraid to talk here. So go ahead.
Right. From all the things I know, right? So theta is super dominant throughout hippocampus and entorhinal cortex. It's a very powerful extracellular signal. So if you
just stick a voltage probe in there.
Stick a voltage probe anywhere in entorhinal cortex and hippocampus. The number one thing before you record any spikes, you will see like these, rhythmic oscillations, right? So it's just a powerful extracellular signal, and of course, that those signals come from somewhere, and they come from the fact that neurons are oriented, and that when they spike, they produce an electromagnetic field. If you have enough neurons that are oriented the same way, then you
When you
get...
say oriented, what do you mean oriented?
Okay, so...
Just firing together.
Okay,
Is that what you're saying?
neurons move charge, right?
Yeah.
When neurons move charge down an axon, for example, right, then you produce a field outside of the neuron.
Yeah, you're talking about spikes, right? So if a spike goes down something,
Right. Exactly.
as the spike goes down there's a temporary voltage reversal. Or
Right, and if neurons were randomly oriented, all of the fields of these different
Which
contributions
is
would
oriented? Weren't you going to physically oriented?
physically oriented. If the axons of all the neurons were pointed in random directions, 3D
Yeah.
spatially, then the total summed field would be very small because all these field effects would cancel each other out.
Why is that? If they were all firing at the same time, it doesn't matter with the orientation.
Oh yeah, you could have like a global up and down regulation of activity, but that's not necessarily what we're talking about when we're measuring extracellular.
But I don't
field.
get it. Already I'm confusing here. It's like, first of all, we don't see axons all oriented in the same direction. We see them going all over the place.
No, not true. Hippocampus and adrenal cortex are highly structured.
When they reach their destinations, they go all over the place. I mean, that's they're branching out in every different direction.
Well, I mean, that's very dominant fiber paths, right?
Okay.
Like
So the question here is, so I got these fibers. You're saying the matters of this, right?
Right.
But the speed at which this information travels here is much faster than this 8 to 10 hertz we're talking about here, right? So these, in some sense, in a phase of a theta cycle, the propagation delay here is insignificant, I think. So basically, I don't see why the orientation matters. The entire field around here is going to get polarized or depolarized, and
Right.
one right here and one right here. And so if they all went together, then they'd all get polarized and depolarized. But I don't see why it would matter. I'm just questioning. I don't see why it matters if the axons went that way. It would still have the same basic big field of depolarization and polarization.
Could
So
clarify
these-
something on this, or when you're talking about the theta, are we talking about a modulation of the spiking rate, or are we talking literally there is a waveform that you can pull out?
Literally, is a voltage. If you just stick your probe not into a cell, but in the space between a cell or anywhere around cell, you'll see this voltage going up and down.
Okay, so what you're talking about is a coherent phase among a bunch of antennas. That's where you're trying to move the
There's an
charge.
extracellular field, right? It's,
Right.
which we call the local field potential, which is the sum of some...
It's almost like you could put two electrical plates on top of the tissue, and you put a voltage across them, and then you'd measure that voltage.
I guess the question I'm trying to get at is, or understand is it that a whole bunch of cells are firing together at the up phase, or is it just that they're all firing randomly and it's this orientation that's causing this theta phase?
And it
And it sounds like orientation may not be the issue.
There's a lot of
What you
different
said.
threads to this conversation already, so I can only break it down a little bit.
We're just trying to figure out what the ups and downs
Yeah, I
are.
understand, but like this, we're already confusing different parts of. So one thing is that local field potential itself is, of course, depends a little bit on your definition, right? So you could say it's the summed activity of some 10,000 neuron, or if you have like a microelectrode, then it might be less than that. But the point is, what you are picking up is not just like the sum of spikes, right? Neurons are moving charge whether or not they're spiking or not. Even in a completely silent network, you would, there'd be stuff.
Yeah, but it would be
Going
static. It wouldn't be dynamically going up and down. There's a constant trickle charge going on.
No. You have lots and lots of sub-threshold dynamics in neurons, right? All of these oscillations, for example, that are described to be intrinsic membrane potential oscillations, are all below threshold.
Yeah.
So if the neuron is not doing anything, if it's not spiking, it's still oscillating. So the point is, yes, spiking has an influence on the field, for sure it does, and we can pick it out. That's why we do spike sorting, to find which neurons spike when we have multiple electrodes, like in tetraodes and whatnot, right? That's how we started recording spike, because it's hard to patch clamp cells. It still is. But you get field effects without activity. That's not the...
So this is an area that I'm not familiar with, and I think it gets to the key of the problem I want to get to.
Yeah.
Because I'm also asking about, they're acting as they say, well, there's some evidence from 1998, and I didn't read the paper, that there's these separate oscillations going on in the dendrite. Well, from what I know about dendrites, there wouldn't be some oscillation over the entire dendrite. If there was something like that, it would be over a dendrite segment, because the segments are sometimes electrically isolated. And so this model of a single sort of oscillation of dendrite doesn't seem right to me. And so it gets to this question. You're telling me that even just the soma itself has this inherent oscillation that's going on all the time.
That's what is called an intrinsic membrane
Yeah, so there's
potential
intrinsic
oscillation, an MPO as it's
Yeah, so they call it there.
But is that what's causing the theta? That's not what's causing theta cycles.
Well, is it? Well, why wouldn't it be?
I don't know, is it big enough? Is it such a
Alright.
dominant
Okay,
thing?
it's-
Like that's the only.
Can we like chop down these points in order?
Sure, but yeah,
We're still not done
yeah.
with the orientation thing. So, in order to properly describe the local field potential that you get from an account of spiking activity, you need to be able to understand how all of that spiking activity influences a local field. If you're
I'm
summing
sorry,
the
you just said
activity,
spiking activity. I thought you said this is not related to spiking activity.
I am saying this is complicated. You are
Okay.
oversimplifying it to a degree that does not, wouldn't do justice to the biology of it, and certainly doesn't do justice to this. So in order to try to explain an extracellular potential like that, a local field potential, right? Colleagues of mine have done this for the neocortical column. So, because the problem is that these investigatory techniques that we have are not compatible, right? You might record spikes, or you might just have a probe to estimate the local field. And something that people have been asking for a long, long time is how can we convert one into the other? What does one have even to do with the other? Just like people are trying to find the link between the LFP and the fMRI signal, right? These are different things. And one of the things that is most important to try to understand the local field potential in cortex is the fact that we have a column and that the column is oriented and that the charge moves along one direction, because that amplifies the total field effects from all of these neurons into a signal that a probe could pick up. That signal would be much, much smaller, right, if you didn't have the alignment of axons in a certain direction. That just to explain the orientation thing.
I still don't understand it. I'm an electrical engineer. I don't understand. I'm not saying you're wrong. I don't understand it. I don't have enough information why that, what's going on there, that alignment is important.
Okay, 10,000 neurons are firing at 1 hertz. So there's 100 neurons active at any given moment in time, right?
Yeah.
So these neurons are actively moving charge. If they were moving charge in a random direction, then the field effect of those charges.
You say moving charge. What you have a process coming from a neuron, and I have a spike traveling along that process. And as the spike travels along the process, there's a transient voltage thing. Now that's if that's what you mean by moving charge, it's not going to be moving charges. There's a moving voltage gradient that moves, but it's not actual charge. Right. There's no current flowing in that direction.
No, but like to an extracellular probe, right?
Oh, that's just be very precise, because I'm thinking in terms of the electrical engineering terms. So there's a transient voltage that occurs at some point, and that transient voltage differential moves along a section like this. Okay, got it.
So you're saying because they're all oriented in the same way, whatever measurements are just amplified, are larger than.
That
doesn't explain the
I
theta.
just still don't understand that.
No, that has nothing to do with theta. That is very basic electrophysiology.
I'm still missing that basic electrophysiology. Okay. So I've got these voltage gradients are traveling along these things. Are you assuming they're all traveling in the same direction, like from lower layers, upper layers, something like that? Is that, or it doesn't matter?
I don't know. I just know that people pay a lot of attention to the morphology.
Okay, so let's say we have a bunch of these spikes all traveling in the same direction, and so you have a bunch of these voltage transients here. Now, obviously, if they're right next to each other, and these cells are firing synchronously. Now, you didn't say that. If they're firing synchronously, then clearly the local effect here would be amplified locally, right? Because they're all all these spikes would be traveling the same. But those are the assumptions. The assumptions they have to have all these spikes have to be occurring at the same time, traveling in the same direction. In that case, I can see why the alignment makes sense. But that's a pretty big assumption that's happening, that these things are spiking at the same time and in the same direction. But if that's what you meant, then I can understand that.
Again, I'm not an electrophysiologist, I'm not an experimentalist, but I do know that the morphology and orientation, for example, the alignment of columns, which tend to have a lot of vertical connectivity because of the microcircuit, is crucially important in
Okay, I'm
trying
trying
to
to
reduce
understand why.
a local field potential from the activity of some 10,000 neurons.
I'm trying to understand it from the point of view of an electrical engineer. Okay, so I gave you, maybe you don't know, that's fine, you can say I don't know. I'm saying as a loss of I don't know. But from what you said, the only thing I can imagine would cause those things to matter, the alignment, just going back to that, is that what you're really doing is you basically have to rely on the fact that these spikes on these different axons are not occurring randomly around here, but they're somehow moving together in a group. And then if they were occurring randomly, then there would be it wouldn't matter which way these things are aligned at all. That's my
I'm
understanding.
still not. I still don't agree with the premise that this is all about spikes.
Well, I'm just reacting to what you said. So,
Yeah, I understand.
So, but if you're telling me that I don't know of anything else that travels along here if it's not a dendritic spike or somatic spike. I just don't know that. So, is there something else that's traveling along there?
Well, I mean, no, but yes, but no. So there's lots of electrophysiology that is just a lot longer than 1 millisecond, right? There's after hyperpolarization, there's ion channels opening and closing, there's all kinds of facilitation and adaptation and depression and whatnot happening. And all of those have an effect on the field because they're changing.
Some of those are related to spikes.
And
some of them are not related to spikes.
Some of those are related to spikes. Some of those are
So
not
if I'm
related
looking
to
for
spikes.
the after potential whatever of a spike, yes. But if I just look at a dendrite sitting there or an axon, you want to talk about axons. If I wanted to say there's an axon that's sitting here, my understanding is an axon that's not propagating a spike at any point in time has an active sort of current maintenance thing, but it's not oscillatory at all. And there's no traveling anything. It's just locally the ion pumps are working, keeping the voltage differential the same. That's my understanding. It could be wrong, but that's my understanding of what goes on. And the only time there's anything traveling or that anything that actually has any oscillatory or even a transient change occurs when there's a spike. So if there's no spike, then there would be no transients and there would be no possibility for oscillations. That's my understanding. Is that incorrect? Or do you not know?
I don't know.
Okay.
Let's assume for a second that it's not a spike.
What is it then?
What if you, just hypothetically, you have charge transport in and out along the dendrite to maintain that homeostasis?
It is. That's what I'm saying. But that's this way. It's local. It just happens locally.
Right. But the thing is that, I mean, what are you picking up with an EEG? You're picking up a summed field potential, right? Not necessarily spikes. So what we're seeing here potentially is the fact that there could be, all you have to do is raise the potential and lower the potential along the entire axon and you get this effect.
Yeah, but I don't have any evidence for that.
Well,
I just
okay, so here's the thing. If you're trying to maintain a cell at a particular voltage level, whatever the basal is,
Yeah.
and you've got pressure to move sodium ions around, potassium ion around, chloride ions around, you could just assume that once you get to a homeostasis, everything remains locked and static. But is that really the case? Are there dynamic processes?
I guess I'm saying everything I've ever read about this says that it is static or stasis at that situation. I mean, obviously, at a moment-to-moment, each channel, ion channels are opening and closing locally. Mm-hmm. But I've never heard of any coordination among them over that there's a traveling wave of that or anything like that. Okay. Maybe there is. I just have not read that.
Okay, so let me put a little more weight on that. You're basically pulling these ions from your local reservoir, right? From the extracellular material, fluid. Okay, so if you pull it in, that's going to create a deficit. Something has to flow in by osmotic pressure to compensate for that. If they're all doing this thing, you could be creating some low subthreshold flows because you don't have an infinite reservoir out there. Everything.
But all right. I agree, Kevin. There's very speculative here. I mean, there's huge decades of research on this.
No, I understand.
And so there's going to be an answer to this question. I think we could speculate. I guess I don't want to do that. I want to say, look, either it's known or not known, and if it's known, either it exists or it doesn't exist.
Okay,
This is important. This is what I wanted to get at here.
I was making a thermodynamic argument, but you can.
I think that's, it's too speculative at this point. It's like these things are very complex, and yet they've been highly studied. So
Probably hard to
I wonder if we can go back just to the original question, what is
Yeah.
the origin of the theta? Like,
Right.
why is it
Right. So
10 hertz and whatever, why is it
yes.
synchronous?
So there are cells that are intrinsically bursting at theta. There are cells that have intrinsic membrane potential oscillation subthreshold. Not necessarily related to
So
activity,
let's talk
but
about which
rising
cells
activity.
are those. Is it like every cell, or is it just like some few cells over
If you
here?
measure cells in entorhinal cortex, I don't know the exact numbers, but I think like in layer 3, there's like a lot of theta cells, and I think that like at least all the stellate cells that are known like are often, the vast majority of them have intrinsic membrane potential oscillations as far as I know. And there's lots of bursting cells that have activity that is modulated by theta, whether that is by subthreshold membrane potential oscillation or by some synchronization of the.
So let me just, because I'm not aware of this. So this is an interesting new thing for me, and I don't know if these exist in the cortex or not. But you're basically saying there's a bunch of cells that intrinsically just have this subthreshold oscillation around theta frequency.
Yes.
And so that's the key thing. I didn't know that. I mean, I know they talk about this here, but I have not read any of the papers on that. So I'm not sure if that's something that exists in cortex, and or did, and I missed it. I don't know.
Actually, I'm really interested whether, for example, stellate cells in layer 4 that get input by cells that would be that would fit the oscillatory input model, so like from anterior thalamic cortex, where there's neurons that obey that rule, whether those stellate cells have an intrinsic membrane potential oscillation in any way.
So if we have an
really
intrinsic
interesting.
membrane potential oscillation, obviously that's measurable outside of the cell, just subthreshold. And obviously if I have a whole bunch of cells together, one can easily imagine how they get synchronized.
Right, although the interesting thing is that, as far as I understand, the intrinsic membrane potential oscillation is not actually synchronized to the global theta, which is more of an activity theta throughout, at least in the hippocampal entorhinal cortex.
So that's weird. So now we again don't know where the theta comes from.
So
Right,
you say
but
this-
again, this is just one model, and there's plenty of models that do this effect without an intrinsic membrane potential oscillation by simply having two oscillators in opposite directions and they're coupled in phase.
But you said the background theta is due to activity.
So there's lots of cells that have theta modulated activity. Yes.
But
They
that's,
call them
that's
there are
but
more
that-
cells firing
but-
in the
but-
up-
But that doesn't tell you anything, because they could be firing because they're surrounded by a theta. I mean, that's how I always assumed it. There's some
Right.
sort of background oscillation, and then the cells are going to likely fire on that oscillation, which this whole model is based
So
on.
there's lots of cells that inherit their theta from cells that are theta modulated, and so they look theta modulated, but they're not actually theta modulated.
All right, I want to get off this topic.
What would synchronize them so precisely? What is causing this?
I don't know. I guess I want to get to some other questions. Can I just table this one? So I'm going to walk away from this saying, okay, I've learned something today. I didn't know that there's these intrinsic theta oscillations in the cell's membrane potentials. I'm not sure if that's over just the soma, over the entire neuropil of the.
It's recorded in the soma. You can probably pick it up. Like.
No, the question is does it propagate through the whole cell or not? Because this requires that there's different theta oscillations. So this
Not
is
necessarily. It could just be bursting input at a theta frequency. It doesn't need to be. You
You're
always.
saying there's some cells that have an intrinsic theta.
No.
Didn't you say that?
I want to. You always say that we're talking about a dendritic oscillation here. That's not necessarily what we're saying. All that we are saying is that the input that comes in through the dendrites is modulated by theta, such that, for example, it might
Yes, I
spike
agree.
at a frequency.
Yes,
that,
I agree, but that's not what this paper said. So that was one of my things
Right.
about the paper. The
That
paper
is-
said, all right, let's go back. The paper said there was an oscillation here and that this could be separately controlled from this. And I said to myself, well, is that really true? And they said they gave reference one paper. I didn't read it, but I said there's lots of other reasons why this could have a different oscillation here other than it's intrinsically oscillating.
Right.
So that was one of my questions.
Right.
And they make the assumption in this paper that that's the only thing they talk about. There's a
Right.
bulk
Can I respond to that?
Sure.
Cool. All right. So the hybrid model that I presented, for example, has inputs from these velocity control oscillators built as ring attractors, which are actually not, oscillations. They're just, spikes that have a theta modulation to them because the activity goes round and round in these ring attractors, right? And so, so you've already seen examples of a model that is not a requirement of the model per se, right? This doesn't.
No, I understand.
You always talk about an oscillation. It doesn't need to be an oscillation. It needs to, it needs to have a theta modulation of some sort, and yes, that needs to be at a slightly higher frequency than.
So that is an oscillation. I'm
Right.
just saying that's.
Right. Okay. Good. But then we
But
agree.
it's not like the dendrite is oscillating, it's the inputs to the
Exactly.
dendrite.
Yeah, I understand. That's one of my points. I'm not arguing that this doesn't make. I'm not sure if this makes sense to me or not. And so that's what I was asking. I'm saying like,
Right.
oh, well, that's a weird thing. And by the way, I don't think it would look like this. And by the way, if their idea that this dendrite would all be oscillating at the same frequency doesn't make sense to me because what I know about dendrite says that wouldn't happen. So, so I was questioning this. That's my question. I'm not arguing for this. I'm saying I don't believe this.
Right.
So-
okay. So the second point that you said about the, whether those cells exist or not, right?
What cells?
The cells that provide input to the dendrite there, right? The cells that are the root cause for the, for an input that is slightly faster than theta.
The ones that are oscillating the
Which is-
dendrite?
Right, which are have a cosine tuning with respect to the orientation and linear dependence in their interspike intervals with regard to speed, right? So this what I described last time as the VCO law, right? So this requirement that there must be input cells that have firing in the theta range. That firing increases in its the interburst interval, right? Decreases with velocity, and it is cosine tuned with regard to speed. Those cells, exactly those cells, have been found. That is the Welday 2011 paper, and I think I even showed some.
Where are those cells again? Remind me.
Some plots. They recorded them from various brain areas. So they found them in, these have been found obviously in anterior cortex, but they also exist in, here they found anterior
Are they,
thalamus.
so in entorhinal cortex, are they specific cells or is this like a general property of cells? That's
The speed modulation?
yeah, it's or this. I mean, they make the point. I think in this paper in the very beginning, they briefly mentioned this that they're introducing a whole new way of thinking about how neurons work, right? And I agree with that. And we did too. We introduced the whole idea of the dendritic spike as a predictive signal and a network effect of that. And this is another one which seems to me really important now, and I'm trying to reconcile the two, right? So we have a model that says dendrites are very, very complex, and they do these local pattern recognition things going on, and I imagine somehow we have to bring these things together. So I'm looking at saying, well, a real dendrite looks, very complicated, and each of these are sort of integration areas, and anywhere along here, every 40 microns could be an integration area, and then perhaps these integration areas are working, independently, are doing what one of these things is doing, So there's a Uber model here, it seems to me, that I think we're getting hints at.
Right, which is why they make an anatomical argument that those stellate cells in medial entorhinal cortex layer 2, which we are looking at here, right, those are those grid cells, have, 4 to 8 prominent dendrites.
That's a separate question. Because
Which would serve as that integration zone.
but the prominent dendrites are all going to have these guys too. I mean, a prominent dendrite just means that at the soma, there is a big trunk coming off here. But then that splits always. I don't know of any cells that doesn't split like this. So to say there's 4 is incorrect. It's like, well, there's 4, but each of these are divided up. It's very, very unlikely that all of these regions here also somehow oscillate at the same time, or they're sharing this oscillate. I don't think that's likely.
So you have an issue between the segments and the dendrites that.
Yeah. Basically, they're not. The word dendrite segment never appears in these
Yeah.
models, and we know that they're really important. That these, in some sense, these guys are almost like electrically isolated at these junctions. I want to keep going. I don't want to spend too much time on this. So I personally, maybe you understand this, I personally need to read more about these things. it's very critical to me whether if this is a generic property of all cells that they have these membrane potentials that are part of their calculation, and it might be every cell is doing this. I was not aware of that, and that's like a big thing. If that's a really important
I don't
calculation.
think at least in neocortex, I'd be surprised if the vast majority of cells had an intrinsic membrane potential oscillation.
Why would you be surprised?
But as far as I know, that's not a property of pyramidal neurons, for example.
Okay. Yes, you're right. I was not aware of it, but I wasn't aware of it actually anywhere. So,
Right.
also, it could be one of those things that have they looked for it? Do they know this?
I mean, plenty of like neurons that are described as intrinsically bursting, for example, are known to have intrinsic membrane potential oscillation, which are might be the cause for the intrinsic bursting actually.
Yeah.
Because it periodically raises the lowers the threshold for the inputs that you need to get spiking, which means that you're gonna get spiking in like these nice little burst packets,
Yeah.
right? And that gives you intrinsic bursting characteristics.
But it's been well known that there are oscillations throughout the neocortex all the time, and so there are intrinsic oscillations going on in the neocortex all over the place, and at different frequencies. And so the whole idea that what role they play, we've never really had a good understanding of it. And now here, for the first time, we have clues as to a computational principle that could be
A
applied.
model that leverages those.
Yeah, that requires it. And
Right.
so I guess I need to find out more information about that. I want to go into a few other things here. There's another interesting thing that occurred to me is like, okay, so you got this phase shift that you're going through here, and I think that's really a better indication of your location than the actual spiking.
Mm-hmm.
And so the question is, do we think that the neural tissue weeds that out? Meaning, for example, are there some populations of cells that are saying, oh, we're all going to respond to some early or late phase shift, and then another population of cells are going to find some early phase shift.
Yes.
This reminds me of in the hippocampus, there is this idea that you're moving through populations of time through the theta cycle, I believe.
Mm-hmm.
So is that a general principle that we think is going on, that as we learn things or as we recognize patterns throughout this cycle, is to the extent that this kind of phase shift occurs, that there really are populations of cells that are saying, oh, I'm here, and another population of cells says, no, you're here, and another population of cells says, no, I'm here.
Mm-hmm.
Is that the general belief?
I think the way that a lot of theoreticians are thinking about this, just to make a very quick example, if you had a cell that did not have precession, and of course there's lots of cells that are just straight up theta modulated.
Yeah, they make a
You
point
get
that
a
here
spike
also.
exactly
They
at
say in
the
layer 2 and layer 3 in the endorhinal cortex, you find there's some that are short precession, some that don't.
And then if you had a mechanism like this for coding inside the field, that would mean you would now get, small, very small time shifts, right? These delta t's, right? In the middle here it's 0, obviously. And these delta t's are very small. And that puts the distance and the timing in the window of an STDP kernel, and thus relationships can be learned, which would otherwise be impossible because this is on a behavioral
Relationship.
time scale.
I'm sorry, you lost me.
Okay, the problem is that passing this thing here, right, takes, I don't know, maybe it's 400 milliseconds, right? Let's assume it's like 4 theta cycles, something like what you drew here,
Yeah.
right? So like 1 spike here and 2 spikes
Well,
there.
it could be longer than that. I mean, the animal could just move
Yeah, depending
along.
on the speed and depending on the size of the
Yeah,
of
400
the
milliseconds
field.
seems pretty fast to me. That's like less than half a second, and
Yeah.
we're talking fields that are actually the size of a rat. So
Fine, make
No,
it
well,
700. I mean,
well, don't worry,
Right.
it could be several seconds.
all right, good. Okay, so we got.
All right, I think
cycles.
I want to make sure we cover that.
Right. Okay.
Yeah.
Good. The point is that is a time window which is way longer than what has been observed in SDP kernels. Right? These curves that you have seen
Yeah.
about potentiation.
Yeah.
Potentiation.
Yeah,
Sorry,
yeah,
my handwriting.
yeah.
Depression.
Maybe I can jump it forward here,
Right.
Florian. I don't have a problem with the mechanism for this. I'm not asking how this could occur. I understand
Okay.
how that could do it. The question I'm asking, do we think it's actually happening? Do we think that there are populations of cells that are responding specifically to this, and another population of cells responding specifically when the animal's here? I guess that's just, I've always felt like, oh, when I'm in some place, I can associate that place. Like if I have a bunch of place cells active, I can associate with a bunch of grid cells, and I can say, oh, I can learn that correlation. It never occurred to me that as we're moving along in this distance of travel, however long it is, that there's going to be a series of locations and a series of place cells that were, it's the same cells but a different,
The exact spike timing lends additional importance to what the firing rate timing signal would give you. Right? The firing rate tells you somehow, okay, am I at the firing field and am I, at the center or at the very outskirts? This tells you much more because it now gives you a much more qualitative.
I got it, but the question is, how is this? I mean, we're looking at the same cells firing throughout here. They're just firing at different times relative to the theta.
Right.
And so the question is, if I think about it, I have another so I have a population of, let's say, place cells that are active at this time, and they're all exhibiting this behavior. And yes, so when I'm here, they're not spiking as much, but they're all phase shifted to the theta. When I'm here, they're spiking more, and they're not phase shifted. When I'm here, they're spiking less, and they're phase shifted the other direction. And I have another population of cells down here, let's say grid cells, that are maybe doing the same thing. The question is it possible to actually recognize this location as separate from this location using these cells? It seems to me I can't. It seems to me like it's the same set of cells active. It's the same set of cells active. They're going to be active at the same time. Yes, they're going to be a different rate of spiking. So it doesn't. I don't see how I could encode or have a population of cells that responds differently here than here. It's the same population. It's.
I don't understand. It's the same cell.
I know it's the same cells. It's always a set of cells, right? So
there's additional information in the exact.
The additional
time.
information is only in its relative timing to the theta cycle.
Right.
The question is, I think, could there be a downstream cell that's looking at this population
Yes, of
and
course.
responsive
Why
to
would
one
that not
versus one responsive to one versus the other?
Right. So you would need a cell that reads input from the green and the black, right? That gets the theta time shifted signal as well as the as just the regular theta signal.
But it would not fire if it was black and then green. It would only. It's got the same connections, but it would only fire when it's green and black,
Right.
not black and green.
Right.
Right? How would that happen?
Well, that goes back to the.
Right.
No. That would
So
just says those two gets associated. It doesn't say that green followed by black will have a different
Well,
response
but
than black followed by green.
Um,
This is a learning mechanism.
yeah.
No, that's causal. It's directional. So
The learning is causal.
Yeah.
So
Yeah, but once the connections are there,
Yeah.
right?
Yeah.
The order of the firing is not going to change the response
Yeah.
of the cell.
I don't understand. The firing is causal from green to black or from black to green.
But Cyril is pointing out that this is a
Yeah,
learning
yes.
rule. It
The
doesn't.
potentiates only in one direction.
Yeah, it's
Yeah,
a learning
exactly.
rule.
So
though.
so assume the learning has happened causally, and now it's got a connection to the green cell and it's got a connection to the black cell.
This is not an activation rule. This is
It's
a
not an
learning rule.
activation rule.
So basically,
Now those connections are there. Now
Yeah.
how is it going to discriminate a green spike followed by a black spike
No,
versus
but-
a black spike followed by a green spike?
Okay, but no. Okay, maybe we misunderstand each other. Right? At least in this little drawing that I made, right, there would be like two impacts, right? One depressing and one potentiating, so that wouldn't actually be a change. Like this would be so let's see, this delta t is negative, so this would be depotentiating, this wouldn't do anything, and this would be potentiating.
Yeah, but those are all learning rules.
No, they're
Those
all
are
learning. So let's say
assume the learning has happened.
let's say the learning has happened. I said, oh, this is. I want to learn this
So you first
relationship.
depotentiate?
So, no. I've learned this.
Potentiate
I've learned this, right? I've learned this. I said, oh, this is good for my learning rule here. This is what I want to see. This one precedes that one. That's what I want to learn. Now, you form that synapse. Now, there's two synapses there, so those two things right next to each other. Now, those two synapses get activated here. This rule says, oh, I don't want to learn that, but the two synapses are there, and as far as we know, there's no rule that says they don't
Would you mind
stop.
drawing that circuit? I'm confused.
The question is, I'm sorry. The question is, you've got some axon or some dendrite and an axon coming down here, and the question is, oh, there's a spike on here and there's a spike on here, and do we potentiate this synapse or not, right? So let's say yes, this is the right relationship here. This precedes this. The green precedes the black. That's the right relationship we want. So this is the green. This is the black. This is causal predictive of this. So this guy gets strengthened.
Right.
Great. Now, a moment later, I get a spike on the black and a spike on the green in the opposite order. That would say, oh, I should decrease this synapse, but it does not decrease the activation right away here. It's basically saying, you know, this green guy is still going to activate the black guy. It's still going to it's still going to depolarize the black neuron. And so just because I don't want to learn that doesn't mean it's not going to have the same effect. So if I was recognizing this pattern, I'd recognize it here too.
Right.
So I wouldn't have a different
Yeah,
cell
so the
population.
activation would be the same in both cases.
Well, I mean, of course, these STDP kernels only come about because of averages of like, you know, some, you know, 1,000
Okay.
spike pairs.
Yeah,
Right?
yeah,
So
but-
it's not an individual spike
I
pair.
know, my point
No,
is.
no, no, but this is like after learning has happened.
Yeah.
The SCDP is the learning process.
Yeah, I still don't follow. These are individual spike pairs. These are learning rules that have to do with what happens once you've applied 100 or 400 spike pairs. right?
Exactly. Got it. So let's say here's a dendrite, here's a black cell, and after hundreds and hundreds of these things, now you've learned
Right.
these two synapses.
If you reliably activated them.
If you reliably
Positive
activate it
temporal
there,
tension,
and it's
yeah.
learned and it's supposed to be specific to this.
Right.
So now hundreds of trials, it's already learned this.
Mhm.
Now how is this neuron? So this neuron, you want it to fire when it's green followed by black. Right? That's what it means to be specific to this.
Mhm.
But if this guy fires first and then this guy fires, you don't want it to fire.
Mhm.
What is the mechanism for that? That's nothing to do with STDP.
Right, that's true, yes.
That's the
Right.
question we're asking.
Okay. Right.
Is there an answer?
That's
That's the question Jeff was asking. Like,
Yeah.
how do you discriminate one position from the other? Not how do you learn
Yeah.
it, it's
Yeah.
how do you actually fire the neurons
Yeah.
separately.
Yeah.
Okay.
It's
So there's no
basically
answer.
an information coding question I'm having here because I have a model in my head of how populations of cells activate other populations of cells.
Now,
And
there are dendritic mechanisms where kind of if this guy fires before this,
Yes.
it's more.
Yes.
So there's
Yeah,
a
so
temporal
there's
thing,
that
but those are really. fine. They're not-
that could be this here. We know that if you have a distal dendritic segment, it's more effective if it fires before, I think, the more proximal dendritic segment. Did I get that right? So they sort of sum up as you go along here. So it's possible that it's quite
There's a
possible.
directionality on the
There's a
dendrite.
directionality on the dendritic segments, and this is like a pattern recognizer, pattern recognizer, pattern. You could form the same. You could form. I have to think about this, but the point is, these guys are shifting in time. These are shifting in time. I just trying to understand the overall coding scheme here, and there's an idea that there's an additional ability to differentiate these things, which we haven't really thought about. I want to go on to the next. I'm not getting the answers I need, but it's okay. I didn't think I was going to get answers. I wanted to have discussions. I'm going to bring up another thing which you mentioned a second ago. They point out in this paper that in layer 2 in the entorhinal cortex, you see this phase precession in the neurons, but in layer 3 they're phase locked. You don't see that. The neurons always fire at the peak of the theta cycle, right? And in layer 2 they fire off the peak of the latest theta cycle. Does anyone have an idea why that would be? Why would you want one set of neurons to be always locked and another one to show this phase precession? It seemed like a weird thing. It was
For
like,
example, because you might want to be able to measure the difference because it's informative for location, as we talked about.
okay, so we can measure the difference. I'm trying to
That's
think what
an
that
idea.
means in neural mechanisms. So now I have a set of cells. These two sets of cells are, let's say, for example, representing the same basic thing, this location. But one is going to be slightly shifted than the other one.
I don't know, as far as I understand, most of the grid cells are layer 2 entorhinal, megaentorhinal cortex stellate cells.
Well,
So
I'm just talking about these other two
There are
layers.
both. There are second 3 as well.
Yeah, sure.
But there's phase precession in layer 5 as well.
Right?
Yeah, the question is, how would I take advantage of this? And, obviously now there's a difference between these two, and the question is that difference important, and what could I do with that? I mean, so yes, now you might have a population of cells that let's assume for grins that they're representing the same basic thing, like a location or something like that. And now one population, it's a population now, is firing right on theta. So it's basically firing right at the center. It doesn't differentiate between these points.
Right.
And the other one is differentiating between these points.
Right.
Okay.
Both of those types of cells exist.
Yeah. So now, how would I take advantage of that? It's back to my other question too, like this encoding scheme. So, so now I have a population and a population that are shifted. It's the same question almost in a sense now.
Yeah.
How do I take advantage of that? What do I do with that?
It's a great mechanistic question.
What,
It's more than mechanistic. It's more of a theory question too. It's like, well, first of all, it's a mechanistic question. I agree.
Right.
There's also like, why? What would I do with that? How would I take advantage of that? Go ahead. Mark.
just one one detail about those. This is MEC layer 2. This is MEC 3, and we'll go like CA3, CA1. One just quick story you could tell about it is that the way this is kind of connected, that goes there. There are connections in both directions here, and of course there's other stuff down here that goes to other parts of MEC. I'm only focusing on one part of the circuit here.
Yeah.
But here, this is I could draw arrows both these ways. Just the quick story I'm going to tell here is, okay, here you have the ones that are always at the peaks. Here you have them, they're like theta locked. In some ways, this seems more like a predicted location, and this seems more like something that has been kind of confirmed.
Why
by
would you
place
say?
cells.
Why would you say it's a predicted location?
Well, it's because it's.
It could occur before the animal enters the place field. It could occur after the animal enters the place field, right?
Fair point. That is a good point. I feel
But
like
let's go with the idea to say what if it's predicted. Let's say, okay, I want you to finish your thought there. If it was moving this direction and hadn't gotten there yet, then it's a predictive signal. It's like saying, oh yeah, how far am I from that thing?
Yeah.
I'm going there, but I'm not there yet. Okay?
Yeah. Okay. My biggest point was like, hey, this diagram, keep this in mind. One of these is kind of projecting one way into the hippocampus. The other is kind of part of more like the feedback loop of once sensory input has been received and everything. That might be relevant to answering your question.
Yeah.
I'm still figuring out the story myself.
Okay, well, I like the idea of throwing in the word like a predictive signal, even though that doesn't quite make sense on the other side here. They did something else in this paper, which I sort of understood yesterday, and I couldn't remember it this morning. They were trying to solve a problem, and the solution to that problem was to assume that these dendritic oscillators were paired 180 degrees. There was a forward one and a backward one. And I can't remember what the problem they were trying to solve that was.
It's one way of getting rid of the need for somatic oscillation, and it solves the problem that the precession would not work backwards.
What else?
It wouldn't work backwards as if the animal turns around or the animal just backs up.
So in the most simplified 1D model, right, with one somatic oscillation and one dendritic input that has inputs that are slightly faster than theta, you would get very nice precession in the forward path, but when you turn the speed around, right?
What about the animal flips around or goes backwards?
Well, that would correspond to the animal going backwards.
No, because it's very much dependent on the head direction.
Right. Yes. Yeah, I don't know exactly.
Okay.
But the point is the model breaks and doesn't account for the experimental finding. They actually put like mice on like little railroad tracks to show that. Where you could type like the mouse gets shipped back on the train while maintaining its heading in one direction. So
Has to go for it.
Right.
No problem.
So there is a problem in the most simple form of model that the precession doesn't go the right way, and that problem you can fix by having two oscillators with opposing directions.
Yeah.
And if you do that, you also no longer need the somatic one.
Yeah. All right. That seems it's sort of a made-up thing, which may be right, but it might not be right. I think to me,
It's mathematical.
yeah,
problems with the model.
the thing I walked away from this, and I'm confirming in here. There's probably a lot of unknowns about this stuff, or a lot of debates about how this stuff works. What I walked away thinking like, okay, we have a dendritic model that matches a lot of empirical data and explains a lot of problems, and it's really kind of explanatory in many ways, and I really like it. But we know it's also not completely sufficient. And now there is another sort of idea that's going on, something that's happening internal to cells, or maybe involving dendrite versus somas, and maybe involving the local dendrite properties, which is another computational property, another computational tool, if you will, another way of solving representational problems. And it feels to me like I want to merge those two. I want to figure out like how it is that this idea of this sort of oscillations and these and shifting peaks, how general a principle of that is of neurons. That's a very interesting question for me. How general? How many neurons exhibit these problems, these issues? And it was just a few just isolated ones someplace. It would be less interesting. But if there's a lot of them throughout the brain, then that's like a really important issue. And then somehow merge that from this sort of very simplistic drawing to more of a realistic dendritic model, where perhaps each of these has got its own little, I don't even want to call it an oscillator, right? Just its own little sort of phase component to it or something like that. Anyway, these are very hand-wavy things. I just felt like it feels very important to me. It feels like this is a big, big clue. This is another computational idea. And actually I'm trying to poke at it a little bit like, what can I represent with that idea? What would I do with it? Is there a series of activations that occur? What problem does it solve, besides grid cells? And that's it. I guess what I really need to do is I need to read more papers. And I just thought it was helpful to have a discussion about it. It gives me a feel for where we all are in our thinking about this.
Can I throw an idea out there?
Sure, go for it. I want more ideas.
What I've been thinking about. So there's this wonderful paper, I mentioned it now, and I actually shown a plot from it once when I did the hybrid model presentation about this this VCO law. So this idea that in order for this to work, you need to have inputs that have a certain firing frequency. So this is some firing frequency f, and that varies around, let's say this is 8 hertz. And depending on the direction that you have, there's some preferred direction, and somehow the firing frequency needs to increase when you're facing in that direction that you're heading in. Let's call it phi naught, right? So maybe this is now, 9 hertz, and then when you're going in the opposite direction, it needs to be less than that, so maybe it's 7 hertz. I think the differences are much smaller. They're like 8.8.
Yeah. Which is what you want. You need to have these. They have to be pretty close because otherwise these firing fields would be too close together.
Yes.
It's really space them out. They have to be very small differences.
Right. Actually, I can take a look at this actual plot here. Yeah, it's like 0.2 hertz difference between the top and the bottom. And the other thing that you need is that this firing frequency, which is normally 8 hertz, it also needs to go up with speed, such that if you go at higher speeds, then it somehow increases. I don't know, maybe it's 8.5 hertz now.
Why is that? Remind me of that.
Because in order to be able to integrate the movement input, you need to have the dendritic oscillator needs to increase with speed. Otherwise,
I understand that. Wait, I'm confused. I'm sorry. I'm not saying you're wrong. My understanding was the pictures they show, the magnitude of the oscillations, the two oscillations were always the same. It's only when you added and multiply them together, then you got this larger envelope that varied.
Right.
So we're talking about, we're not talking about the sum of the oscillations. We're talking about the input on one of these sine waves, and they didn't require that there be a higher spiking frequency.
No, because they analyze that whole model that you build in your head is at like a constant movement speed. The entire picture gets a bit more complicated when this has to be able to work at variable speed. But when you go faster, what's supposed to happen is there's you will spend less time in this field, so you're not going to have 8 cycles.
Go faster. The encoding of speed, I thought, was just the change in frequency.
Yes.
You just have to get faster frequencies.
Yes.
So
yes,
it's not a number of spikes. Oh, frequency. Of course, it's spike.
Right.
Yeah.
Right.
All right. Sorry. Fine.
Okay. Right.
Oh yeah. Okay.
Anyway, so these are like these two requirements of what is called the VCO law, right? And the fascinating thing, since I've been thinking about implementing all of this in the cortical column, right, is that there's stellate cells in medial anterolateral cortex, so there's stellate cells in layer 4 in cortex, and the interesting thing is they get input from thalamus, and what Belda et al. Did, amongst other things, they recorded cells from anterior thalamus, anterior thalamus, and it turns out there are cells here that obey VCO law. So they have exactly this kind of cosine tuning and linear
So
relationship.
we're talking about what we typically call relay cells, you're saying?
Yes, and they're theta cells, so they're bursting.
Wait. When people talk about thalamus, they talk about relay cells projecting to layer 4. Are you saying that's the same cells, or is it a different population?
I don't know that. They just call them, anterior thalamus cells, AT1,
So
AT2.
this goes back to my question earlier, whether there's these specific cells for creating these frequencies. This is more how I always imagined it. I always imagined that there'd be some set of cells someplace. Thalamus would be a logical place that we know that is involved in this, where they are basically the timekeeper and they are sending out some frequency, and it wasn't happening everywhere, but these guys were driving it. So now there's two ways it could happen in the thalamus. It could be that the relay cells themselves have this property, or it could be another set of cells, which like the matrix cells, that are doing this, and they project broadly through cortex, but they're not the relay cells. I would have guessed, if I had just to guess by myself, I would say, it's probably the matrix cells that do this. And I've always suggested, this will come back, I've always suggested the matrix cells encoding speed. I've always said that. That was back to the temporal memory, the melody example. So
Why? Because they sync across.
Because you want to have a universal speed over some set of population of cells.
Well, why couldn't the relay cells do that? They're projecting all across cortex just in layer 4.
Well, because they could do that, but the issue I was getting at is there's this idea that you need to encode time, and time can be independent of the actual sequence. Time is independent of the thing you're encoding. Like a melody is a melody, independent. It's all, but I can speed it all up. I can speed it low down. So I want to encode the melody in one form. I want to represent the tempo in another place.
Yeah.
Tempo scaling.
Yeah, tempo is just, but now I've extended this idea to the scaling in space as well as time with the matrix cells. So, but the idea that the matrix cells, the only thing I could ever find that fit that bill, they anatomically look right, but they do not project specifically layer 4. They project broadly through cortex, layer 1 and layer 6 or something like that. So they're more like a diffuse timing signal. As opposed to the relay cells are very specific. They project just very specific points. So that would be cool if that was the matrix cells, and I don't know what they mean
So
by
all they say here is that they recorded rhythmically bursting theta cells from medial septum, hippocampus, and anterior thalamus in behaving rats.
Okay, I'm going to guess that those are not the relay cells. I'm going to guess that those are some of the other cells, maybe the matrix cells, what are sometimes called matrix cells. That makes sense to me.
Anyways, my broader idea was that these inputs need to be shared, because of course what these cells are really doing is they're integrating in information. And if you apply this more abstractly, if you want to...
What cells are shared by whom?
The input, right?
The input.
The inputs that drive. So what is here? The velocity controlled oscillators, right, which feed into.
So when you point to the thalamus, those are the generator of those are the VCOs. You're saying,
right? So there's cells here that obey VCO law,
and that input has to be shared across multiple things.
My broader question is: this is with regard to movement speed and and direction of travel.
Yeah.
But if you're going to apply this to a broader thousand springs theory idea of working on concepts, so now you're not moving in a physical space, but you're moving in a concept space.
Yeah.
Then you need to have a place that, for one, gets the kind of information from which you can decode the direction of the change, so to speak, like the direction you're navigating in your conceptual space. And the other one is that information needs to be broadcasted in order to have grid cells, because...
That's what you meant by shared. I want to broadcast it so it's shared by everyone.
Right, if you want the grid cells to be in the cell, right, then they need to share this input about that movement and probably over a wide area of things. And so that provides some constraints on the projections.
Okay. So that's, I'm going to go out on that. I'm going to say that's almost definitely the matrix cells, right? And I can explain why. And
Because they go out layer 1, right?
no, it's because they project broadly, not specifically.
Right.
They project broadly within a modality, not across modalities. This is an important issue because the speed at which vision may be occurring and the speed at which audition is occurring are not necessarily tied together.
Right.
But within vision, and the one thing I'm attending to, and the one thing I'm hearing, they're all going to be tied together. So all the columns that are listening to some music symphony are all going to have to work at the same speed, but that could be independent of something else going on visually in my world, how I'm moving my hand over a cup. So you have that. We also have, when I was thinking about the melody problem, there's a need for adjusting the speed based on sensory input. Like if I find the melody, if I'm hearing a melody, I've learned it at one tempo, now I'm hearing it at another tempo, I need to adjust these guys to match the input. So there has to be a sensory correction that basically says, hey, you know what, I'm off here by a little bit, and by being off a little bit, if you speed up, we can be in sync. And so there's these connections back to so essentially a small set of cells that project broadly within a modality and also receive information broadly from that modality. And that was the right location for them, for those cells to exist to exhibit temporal control.
Right. Do they get a copy of the layer 6 modulatory input into thalamus?
I don't remember.
Because I mean that's
When you say-
the prominent modulatory projection, right? Down
When you say
from-
the modulatory, I mean the
What
same cells that project back down to here.
So there is. I'm saying modulatory because of course there are the intrinsic bursting neurons.
Yeah.
In layer 5,
Yeah.
which drive thalamocortical relay cells, right? TRCs.
Yeah.
But these cells are modulated by layer 6 cells.
Yes, I don't think it's those.
That is
What I
the-
think-
that is the-
the data from 25 years ago
Yeah.
was that the matrix cells got input from the other layer 5 cells, not the intrinsically bursting ones.
Oh,
the big.
separate ones.
The other layer 5 cells, those other layer 5 cells, by the way, are voting cells. They also project broadly. But if I recall, it was the layer 5A or the, it depends on what animal because they switch around the animals. But the non-intrinsically bursting cells, the smaller ones, generally that's viewed as two populations. And those are the ones that project to the matrix cells. It's old data. I mean, actually maybe 30 years old, so I don't know if it's still true, but if it's still believed. But anyway, it was a different thing. So it was like, okay, so to me, these cells, these other layer 5 cells, were somehow the signal to tell this guy, like, you know what, you're off, we need to speed up or slow down.
Right.
And also, there was this idea that in music, it's not amorphous. You have notes, and there's an attack on those notes. And music always has that. It's not this. So the idea that if you need to recognize the interval, the internote interval, you need to have some sort of signal to tell you when the note begins. So the idea that you have sort of an attack, and that it's the time difference between those attacks, which is the clue. And so there'd be like, we need some kind of cell here that sort of, it's detecting the time difference between the beginnings of these intervals, at the start of these intervals, and when the next one, like one first attack and the next attack, and somehow those guys are determined, hey, that matches something, I can make this work if we speed up or slow down. It's all fuzzy, but the matrix cells fit the bill in terms of this anatomically. They made a lot of sense. And there's been a whole bunch of other evidence since then. They do project broadly in layer 1, which sort of imply that the apical dendrites on layer 5 cells and layer 2-3 cells are receiving this timing signal.
Right. Okay. That's all great for temporal scaling.
Yeah.
My more abstract question, right? With this is, of course, this is easy to show because like velocity is physically measurable. How would that look like when you're navigating a concept space? Right?
I don't know, but I can bring it outside of audition. Can we come back to that more difficult question later?
Right. Sure.
One of the things that I thought this might also solve is when you're looking at something visually and you're saccading over the object, let's say, and you're looking at where the different components of the object are. Well, the angular movement of your eyes depends greatly on the distance of the object to your to your body, right? So if it's closer, you have to have larger angular movements, and it's further away. It's perceptually, you're not aware of this. I mean, yes, if you ask me how far away you are, I can tell you that. But when I look at you, I'm not thinking that. It's like, I don't say to myself, oh, that's fluorine at 6 feet versus fluorine at 4 feet. It's more like, and so somehow the brain abstracts out that distance in the same way it can extract out the temporal difference of a sped up melody and a slowed down melody. So I think is going on, it was a very speculative idea, but I think what's going on here, the same, you could, instead of encoding velocity of movement, I'm encoding the scale of movement, how far I have to, how far away you are. That if I can scale everything down, if I can say, oh, if I just scale everything down, my movements will go slower, or I'll go less fast or less distance or more distance. I would scale my movements to match the different sizes of the object at different scales.
Right, so it's not really that I'm moving faster in the space, but the whole space gets...
The whole space, that's why I say it's the concept of scaling space, not just scaling time.
Right.
And I think you could do both. I think it's sort of a space-time equivalent system here. Another example I've used in the past...
But it makes it tricky to explain then a cortical grid cell, right? Because the way that it's been observed and described for navigation is that there is a very concrete linear relationship between the speed of these theta cells, or like their frequency, and the speed, right? That linear relationship there.
This could be one of the reasons why cortex is not entorhinal cortex, right? Cortex is completely built around the thalamus. And perhaps when you're navigating a world, you do not need to scale your world. I don't need but when I'm navigating objects, like another example I gave was the coffee cup, right? I've learned the Numenta coffee cup at a certain size. But if you gave me a miniature one, A, I'd recognize it right away, and B, I could manipulate it. I would know how much to move my finger to pick it up, and if I wanted to put my finger on the rim, I could translate all of my movements onto this object I've learned at one size into an object that's smaller. So how the hell do you do that? It's like you have to have a scaler. You have to have a scaler. Everything has to scale somehow. Movements. So anyway, I haven't even thought about concepts yet. That's an interesting question. I'm reluctant to speculate on it right now, but I bet you can give it.
Can I give another puzzle piece here?
Yeah, sure.
So obviously that's one of the puzzle pieces I was thinking about for cortical grid cells, right? Where would that shared input be? And to me, that was a clue that you find those cells in thalamus. But if we don't go looking for this because it's hard and because it might actually work differently, at least the way that you conceptualize it, there's other things we could look for. One of the things that at least the hybrid model predicts that amongst the grid cells, if they're going to be putative cortical now, one of the things that helps them stabilize the metric is the disynaptic inhibition. Right?
Yeah.
The powerful component of the CAN network.
The CAN network.
Right, the CAN network that we added onto the way that we integrate inputs now, right, in order to get stable metrics. And so if that was true, right, hypothetical scenario, you just argue, okay, stellate cells in entorhinal cortex as well as in higher cortex, and some of them might be grid cells. If that were true for the cortical column 2, and putatively, just because I happen to know that there's stellate cells in layer 4, quite some. Is there a microcircuit that would implement disynaptic inhibition amongst layer 4? I'm gonna call them putative grid cells now. Is there a circuit mechanism?
Isn't there disynaptic inhibition everywhere in the cortex?
Yeah, sure. But what I'm looking for is rather specific kind of inhibition now, right? Because what you need is this inhibitory surround profile that says that you're not supposed to inhibit grid cells that have the same spatial phase or conceptual phase, if it's going to be abstract now, right? But you are supposed to inhibit all the grid cells that are part of the.
Couldn't the small basket cells do this? Small basket cells, there are two types of baskets, large and small. The large ones span a lot. They just basically this cloud of inhibition over a fairly broad area. The small basket cells would look the same, they seem the same in some role, but they're much smaller distances. There's not as many small basket cells as large basket cells, but there's still quite a lot. It's still a very popular cell in the cortex. And I think all you want is you want a cloud of inhibition that essentially says, don't go, you inhibit everything around me, but as soon as you get beyond that inhibition, then you can represent the same thing again.
Right.
Which is sort of like the idea of that phase cluster. So remember going back to the tank picture, those phase clusters were, I think, about 120 microns in distance. So you would be looking for an inhibitory cell that maybe has a span of about 120 microns.
Right.
Where large basket cells are much larger than that. But I could
Yeah.
check, small basket cells could fit in me. I think that's all you need, right?
Large basket cells reach like a millimeter to 2.
Yeah, something like
and
that.
small basket cells like a tenth of that or something.
Okay, well, that fits.
Yeah.
Okay, but does it need to be more special than that? It seems to me if I could just prevent anybody else from firing while I'm firing, and then after that they would naturally fire again, and then they would inhibit and so on. Does it need more than that? I don't know if they're in all layers. Because if small basket cells played this role, if they were the role of the can, then you could figure out where cans exist by where these cells exist.
Right.
I bet you I'm sure that's known, or at least there's some data about it, about where the small basket. Now that's a very interesting idea. I like this idea.
Next question. So maybe we call this like the third thing now. If we're gonna, just running complete hypotheticals, right? So if grid cells are layer 4 stellate cells, right?
Yeah.
Is there any known data on intrinsic membrane potential oscillations in these cells? Does anybody know?
I just told you earlier that I'd never heard of intrinsic membrane potentials in cortical cells, so I don't have any data. But I might have ignored it. It might be all over the place. I just never read those papers.
Notably, grids can be built, doesn't need to be theta. Thalamic inputs have alpha modulations, like these.
They have like that.
Right? Again, it might look different if we're looking at the matrix cells, I'm not sure.
Yeah.
But the broader point is there's no reason whatsoever to have to build a grid with theta. It's perfectly fine to build a grid at any rhythm. You can build a grid with alpha or delta waves.
Yeah.
Right? Of course, the rhythm that you choose has some impact on the resolution that you are able to get out of the system, the amount of precision that you need with your phase resets and whatnot. But in principle, that principle of an oscillatory interference regime can work in any frequency range. And if you want it to work in entorhinal cortex, where there's all the theta, well then these VCO law things need to be, around theta frequencies. But pretty obviously, right? Because they need to be very close to the carrier here.
Yeah.
But so what about other frequencies?
Yeah,
right? If there are thalamic inputs that are at alpha, then
that's a good question. Another interesting question is if you find multiple frequencies occurring in the same general tissue, and let's say they're computing different things. Right? They're being, the same principle is being used, but you're using different frequencies to compute those different things. Then they might interfere with each other. Imagine like one set of cells is using one frequency to calculate distances or something here, and another set of cells right nearby are co-located with them or calculating a different thing using a different frequency or something like that. Could they coexist without? There's some level of interference, but they might still work. It's like a union of frequencies. You'll still be able to. The system might still. My point is, you might be calculating multiple things at the same time, using two different frequencies, and all the system might still work somehow. I don't know. I'm just throwing that out as a possibility. Because there are multiple frequencies, and they must be important. You think? Yeah, that's very interesting. The good news is a lot of this stuff can be looked up. By the way, I've never thought of layer 4 as being like the grid cells. That didn't make sense to me. Because they need to be place cells. They're driven by sensory input. They're driven by the relay cell. They are encoding specific sensory input, and
is that true for all the layer 4 cells? How many of them are non-canonical?
There are not many. There are some well, it varies, depends on different studies and places, different animals. But for many people said, oh, layer 4 is all stellate cells. Now people say, oh no, there's a percentage of them that are pyramidal cells. But it's always a minority. I think in the order of maybe 10, 20% are pyramidal cells. And it's not, I don't know of any, other than inhibitory cells, I don't know of any other type of cell there. I think that's just it. And I don't know if there's good studies that show how the pyramidal cells work differently than oh, by the way, that's interesting too. Just think about this. Go back to the tank paper, right? You remember in the tank paper, there was these circles in that image. These are pyramidal cells.
Oh yeah, right. Yeah.
And I
the
asked
pyramidal
him-
cells cluster. For
I
some
asked
reason.
him, I said, why do you think they're pyramidal cells there? He had no idea. I didn't really see them tell me what it was.
Right.
And so I said, that's really curious.
And the weird thing was that the cells on the edge of the pyramidal clusters were then also grid cells, but not in the middle of the pyramidal clusters.
But anyway, the point, if you just looked at this, you might say, oh, there might be 10 to 20% of these cells are pyramidal cells and the rest are stellate cells.
Right.
And I just occurred to me when I remember reading about layer 4, people say, yeah, about 10 to 20% of the cells are pyramidal cells and the rest are stellate cells. Coincidence? I don't know, maybe. We don't know why these are here. I don't get any clue, actually, but point is that would be an indication maybe these are something. Anyway, I've always thought of layer 4 as like, no, these are sensory-driven cells. These are the ones that you would be place cells. They're going to directly encode, where a grid cell has to be activated, in some sense, initially independent of sensory input. It has to encode the location, regardless of what the sensory input is. Then it can learn it. So we've always felt that like the layer 6 cells would be the grid cells, or some of the layer 6 cells, and layer 4 cells, or maybe layer 3, but maybe layer 4 cells would be the upper layers, let's put it that way, would be sort of the place cell equivalent. And then you have these very strong connections between layer 6 and layer 4 that are very canonical and they look very specific. And so that's exactly what you want to find. That's exactly so. And this seemed to me, since these were really driven by sensory input, these would be the place cells.
Right.
And
Which is the
these,
same that you get, you know, in this loop.
Oh, well, let's go back to that
with
again.
with the
I
grid cells into
haven't,
place cells.
these are grid cells to place cells.
Right.
And then, but then this goes to here, back to here. Well, that's okay. That's like, that would be like, this would be like layer 6A to layer 4 to layer 3 to layer 5.
That's a crude analogy, but okay.
Oh, I'm going with it. Okay, right. Because what you find in, I'm sorry, this is layer 4. This is
Layer four, right.
Layer 4. Because that's what we see in the cortex, right? We see layer 4 basically does not project subgranularly.
Right.
It basically only projects to layer 3.
Right.
Layer 3 then projects broadly, but also layer 3 is a very canonical major circuit goes back to layer 5, and
Yes.
I can't remember which part of layer 5. I think there will be some broad similarities, maybe in the 80% range between the hippocampal complex and neocortical circuits.
Once we figure out the circuit for cortical grid cells, wherever they may be.
Well, it helps. You can look for now. They're not going to be identical, right? But they're
Right.
going to be basically built on the same underlying methodology, underlying theme of representing stuff. So there's, the theory we have is that this evolved first, and then it got rearranged into a columnar form, which allowed the cortex to make many copies of it. And maybe one of the other additional things is we got this timing. Another thing to make this work is we had to get this timing modulator. Where we don't need that here. We have to obviously have to encode speed, but we don't have to encode scale. Like the rat doesn't,
Since you have modules at different scales already, prefabricated, right? The modules that
In fact,
get used.
Yeah, in fact, and we also know that if you stretch the environment of the rat, right, everything stretches up to a point,
Right.
and then it like resets. And to me, that always meant that the rat no longer sees the room as the same room.
Mm-hmm.
So if I took this room and you made it shrunk down, I would still recognize this room. I go, oh, this is a miniature version of that room. Oh yeah, and the chair should be over here, and the whiteboard should be over here. Like a little dollhouse version. I would know what it is. But it suggests in the entorhinal cortex or the hippocampal complex that you wouldn't be able to do that.
But there's a breaking point.
There's a breaking point that says, I can't scale. I'm trying, but no, this is someplace different. and I have to start all over again. I won't see I wonder if we could test that in humans somehow.
We could make them giants.
Well, the problem is, I believe, it's interesting. When I look at a room or building like this, I think my entorhinal cortex is encoding its space as I navigate around, but I believe my neocortex is also recognizing this and says, I know what this is, and I can see it scaled up and scaled down, whatever. It's a bunch of objects, and it's a much more rich model. But the thing is, interesting at night when you're walking around in the dark, or in very, very dim light. I do this all the time, you know this, but you can really sort of get a sense for what you know. And so I'm wondering if I were trying to walk around in the dark in a scaled room, would I fail?
I bet you would fail miserably.
I mean, would I be able to no longer say this is the same room? It would just.
I bet you wouldn't match that. You wouldn't anticipate the scaling.
But I would do that if I was looking at the room.
If you were
If
looking,
I was
sure.
manipulating with my fingers.
But perhaps
like
you're
dolls.
onto something about how that's inherently different
Yeah.
as far as scaling goes than objects.
So that's a really nice hypothesis that came out of today. And I think we might have bounced these ideas around somehow in the past too. But the idea is that, okay, you've got another major thing that's going on in the cortex you don't have in the hippocampal complex is this scalar of time and space. Which is not just the VCOs, it's a scalar thing. It's in addition to that, right? It's not just encoding speed of movement of the body, but it's encoding the scale of the object somehow. Like that. Anyway, I don't know. But you got any other ideas you want to throw out there?
Yeah, sure. So an interesting overlap that, reading the Tank slice hypothesis and the Constantinescu paper back to back
What's
is
the time slice hypothesis?
the grid slice hypothesis. So
Right.
So you got the grid, and the idea is that the response on a
Okay. It's not time. I mean, it's the idea that you get the 60-degree variation in the volt signal as you're moving in a certain direction.
No. The problem that the firing fields in a 1D track are not perfectly rhythmic.
But this explains that, right?
Right, and so well, I mean that's the way that Tank explains that the firing fields are not rhythmic because, in fact, what's underlying that is a 2D map, and now you're going at some certain angle through that map, and that's why you see these things. And the ratios between those blips are somehow compatible. It's like this square root of 3 or 7. Forgot the math behind it. But anyways, he tried to show that, and there's of course people who doubt that, but. Anyways, my point is reading that in together with the Constantinescu paper, which had a 2D map of the birds, right, with the legs and the neck.
Yeah.
Immediately makes you think that okay, if there were cortical grid cells for a concept, and now that concept happened just to be a 1D. Given that the representation is 2D because maybe laminar sheet imposes that, just like it imposes that in entorhinal cortex, does that mean that the firing field of a cortical grid cell for a one-dimensional concept would also have
this nonlinear bumpiness to it?
Exactly. And would there be any way to detect that?
How would you detect that?
Would you train people on a one-dimensional concept and you ask them to navigate it? And the question is, are there cells that respond?
Well, you'd have to be careful. You don't only want to train them on because if you're one-dimensional concept, just slide along one of your X and Y axes.
A linear to one-dimensional.
If it lined up with the bumps, then you'd be okay, right? You have to make sure that you have it. You know what I'm saying? You have to make sure that you.
The alpha angle is a freedom here. Right.
That's pretty tricky.
But hold on, one of the main points, both of the Constantinescu paper and the stuff that Moser has done, is to show that the orientation of the grid is not arbitrary. There's a 7.5 degree bias towards the cardinal axis. Is that also true for? Like if I could somehow read out the angle of the grid cells of the Constantinescu paper, would that grid that is behind that 6-fold symmetry that they find also have a 7.5 degree shift from one of the two cardinal axes of legs and neck?
I don't know.
So the place where this came up was another fMRI one done by Josh Julian, and that's with the humans humans with vision, with the way their eyes are facing, and they found something involving the 7.5
Wow.
with how the angles are relative to the square that the person was viewing. And the cool thing was the orientation was anchored to the square's orientation. Or if they took this rectangle,
Right.
the person's looking at a rectangle full of like letters or distractors and everything like that.
Right.
If the rectangle is rotated, the grid that they're inferring from the fMRI
Would
rotates
co-rotate.
with it.
Right.
And
like
they found
navigation
evidence
grids of
that it
the
has
boundaries.
the 7.5
We
degree.
have we reviewed this paper? I don't remember this. Is this new?
Once I summarize a couple of these papers, I can send it
I
out.
forgot. It's all right. But we talked about this. I forgot it. I'm sorry. So this was another fMRI study indicated.
The fMRI indicating that people.
We didn't reference this in the framework paper, did we?
I think so.
I only thought we did the
One of
content
them.
skew and I thought we only did two, the open brain one, surgery one, and the contents skew.
At one point, we were referencing the Josh Julian one as well.
Maybe you did
Maybe we took it out of Metro.
Framework plus, cost plus.
Uh,
Hey, we're gonna run out of time here. That's really interesting. Thank you for bringing it. That's really fascinating.
Right.
Did you want to say more about that?
No.
We're gonna run out of time here. I want to make one other just little side point in here. They were talking about this this idea that these things should be that these different directional things should be 60 degrees was an optimal thing,
Yeah.
and they assumed that's what you wanted.
Yeah, but you don't. need
But
that.
but if you didn't have that, then you would end up with this sort of lumpiness that we see in, as you showed the other day,
Right.
this lumpiness we see in the tank paper. And they did make one observation. They said anywhere down to about 20 degrees of separation seems to be
Right.
just a grid pattern,
Yeah.
right? Now
Right.
20 degrees. If you took 180 degrees and divided by 20, you get 9, right?
Okay.
How many different mini columns do you have to go through to get 180 degrees shift in V1? About 8. so this could be
Yeah.
I just want to point it out that going back to the hypothesis that mini columns are going to try to divide up the space in single dimensional sort of I don't know what the word to put it anymore, but you're dividing up some high dimensional space and you're going to map into a bunch of directional components, and you want to at least have 20 degrees between them. Well, this matches empirically what, by chance perhaps, maybe not by chance, what you see in the video.
One of these days they're gonna, accuse you of being a numerologist.
It doesn't matter what they accuse you of. If you're right, you're right. If you're wrong, you're wrong.
Right, and the
So-
cool thing is, since there is no theory, any theory is a good one, right? That's that.
Well, I don't put my hat on any one of these things.
Right.
You can't. Every time I say well, it could be chance.
Right.
It may not mean anything. But if you find 16 of these things lined up, then you don't think it's chance anymore.
It's hexagons. It's all about hexagons.
I thought that picture you made on last week was very enlightening to me. That that you're going to get just the explanation. You're going to get these hexagonal grids no matter what you do. This still brings up a very deep question for me. Is we need to represent location much more accurately than a grid cell module does. We have to be able to have specific locations on specific objects. We also need to be able to represent states of those locations. We also be able to represent orientation. All these things have to happen. And the question is, I haven't seen yet how all we've been talking about here still is like one grid cell module and how it's going to work. Yet, I don't think that's sufficient. We have to get, and I don't think, and again, I've argued that I don't think you can go down and pick the next scale, the next grid cell module at a different scale. It's almost like somehow we have to encode location out of this one grid cell cluster, phase cluster type of thing.
Right.
I don't know how to do that yet.
Well, there's.
Oh, I have an idea about it. Go ahead. Did you want to say something?
No.
Yeah. Okay. So this is a really fuzzy idea. Okay, it came back to me. So, as we said, if you had perfect 60-degree orientation vectors, you get this perfect grid, and all the grid cells would be properly lit up in all situations. Like you wouldn't see what Tank saw. Tank saw that we have multiple grid cell clusters, and some cells are active and some cells are not active. It's like a mixture of things.
Yeah.
So if you have perfect 60 degrees, all these cells will be active, and if you start moving away from that, then you start getting some dropouts. for the reasons you showed in your
Right.
picture the other day. I have something going like this, and they're not actually lined up.
Yes.
Now imagine what would happen if I added more and more one-dimensional vectors here.
Right.
But I may know it. All I want to make sure is they're somewhat separated from the other ones. But if I'm thinking about a 360-degree space here, and I want a minimum of 20 degrees, well, I can have them all over the place, right? I mean, this point is,
Right.
I can have a bunch.
Yeah.
Right. I can have a bunch of them. And what would happen if I take a bunch of these things representing some higher dimensional space, and I drop them down onto this two-dimensional sheet? Well, I'm going to get a lot of holes. Okay, these things are just not all going to line up. They're just not going to do that. And now, in some sense, that is a different encoding. It's saying like, so think of it this way: the more somehow there's some sort of mapping between this n-dimensional space onto this two-dimensional space, and the result of that is you're not going to get these perfect grids like this. You're going to get holes all over the place. And the question is, how much information can I encode in this two-dimensional space? How much does it tell me about my position in three-dimensional space based on the holes? Based on like where these holes are here. Can I represent enough information by saying, yes, and think of it this way, if I have a grid cell module and every cell was equally active, like I didn't see what Tank saw, that is in some sense a union of all the locations that could be represented here if they weren't all active.
Right.
It's sort of like the equivalent of the mini-column union, but I don't have enough of them. In the Tank paper, he was showing like 6 of these. And so it seems to me the 6 of these doesn't give me enough depth to how many different ways can I say represent 3 out of 6? What's 6 choose 3? It's it's not a big number.
Right.
But it was a hint, and maybe the answer there. It was a hint of like, oh yeah, well, if I don't know really where I am, I'm just going to get all these cells active. But as soon as I actually know where I am in three-dimensional space, then I'll start getting this more sparse representation here, and now I can represent that. And I think that's the end of our time.
Yeah, I think that's the end of
Food
it.
is arriving.
Oh my God.
You can keep talking,
No,
it's just
no,
that I
no,
gotta
no.
eat.
We are live streaming, so.
Well, we're off now, right?
Free-flowing session here.
All right.
That was great.
Right. I have more crazy ideas.
All right, well,
but-
good. Many
More
crazy
more
ideas.
crazy ideas
Yeah.
next time.
More
on,
crazy ideas
the
next time.
Numenta
Are we
Stitch
offline?
Live stream. Goodbye, everybody.